Assalamu alaikum friends. I hope so that you all will be fine. In this video, I am going to discuss about Rapaport Leubering cycle. In my previous video, I have already discussed about glycolysis pathway. If you are new on this channel, then kindly like, share and subscribe to my channel. So let's start our lecture Rapaport Leubering cycle. This cycle was first introduced by Rapaport and Leubering. And this compound was first isolated by Rapaport and Leubering. Students, this cycle it is a supplementary pathway for the production of 2,3-bisphosphoglycerate in matured RBC. It is a supplementary pathway. It is a supplementary pathway for production of 2,3-bisphosphoglycerate in matured RBC. Because matured RBC have no any mitochondria, so for the energy production depends upon the glycolysis, and this Rapaport-Leubering cycle it is the alternative pathway of glycolysis. Okay, students. Now coming toward the glycolytic pathway, how 2,3-bisphosphoglycerate it gets generated. So what happens that this glucose, as we discussed earlier in glycolysis, glucose converts into glucose 6 phosphate, glucose 6 phosphate converts into fructose 6 phosphate, fructose 6 phosphate convert into fructose 1,6 bisphosphate, and this converts into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. This converts into 1,3 bisphosphoglycerate, and then it converts to 3 phosphoglycerate. This 3 phosphoglycerate converts into 2 phosphoglycerate, and then it converts into phosphoenol pyruvate and ultimately pyruvate. Okay स्टूडेंट्स, हम ग्लाइकोलाइसिस में इस चीज को डिस्कस कर चुके हैं, ये ग्लाइकोलाइटिक पाथवे। अब 2,3-bisphosphoglycerate प्रोडक्शन कैसे होती है? This 1,3-bisphosphoglycerate converts into 2,3-bisphosphoglycerate 2,3-bisphosphoglycerate by action of bisphosphoglycerate mutase, and this 2,3-bisphosphoglycerate it then converts into 3-phosphoglycerate with the help of bisphosphoglycerate phosphatase enzyme. Okay students, I'm repeating again. 1,3-bisphosphoglycerate it converts into 2,3-bisphosphoglycerate with the help of bisphosphoglycerate mutase enzyme, and this 2,3-bisphosphoglycerate converts into 3-phosphoglycerate with the help of bisphosphoglycerate phosphatase enzyme. In this way 2,3-bisphosphoglycerate gets synthesized. Okay students, this is so that's why it is called a supplementary pathway for the production of 2,3-bisphosphoglycerate in matured RBC. Students, this 2,3-bisphosphoglycerate is very important. Now coming toward the functions of this Rapaport-Leubering cycle. This 2,3-bisphosphoglycerate it occupies a negative charge. It occupies a negative charge due to phosphate groups. This 2,3-bisphosphoglycerate occupies the negative charge due to phosphate group, and it gets attached to the positively charged amino acid of the beta globin chain. Friends, I am repeating again. this 2,3-bisphosphoglycerate, it occupies a central pocket of hemoglobin molecule, and this is a negative charge containing compound. It gets attached to the positively charged amino acid of the beta chain of hemoglobin. So let's see. This is the beta chain, and here it is a alpha chain. This is a hemoglobin. Students, this 2,3-bisphosphoglycerate occupies a central pocket of hemoglobin. As it is a negatively charged containing compound, it gets attached to the positively charged amino acid of beta chain of hemoglobin. Okay, this is a positively charged. Okay, so as it occupies a central pocket of the hemoglobin molecule, it is responsible for the unloading of oxygen from hemoglobin to tissues. It is a function, it is a very important function. It is responsible for the unloading of oxygen from hemoglobin to tissues. Okay, and the another function is due to phosphate group, it is responsible for the maintaining buffering action. Maintaining buffering action. Friends, I'm repeating again the functions of Rapaport-Leubering cycle. Basically, this 2,3-bisphosphoglycerate compound occupies a central pocket of hemoglobin molecule. And it is a negatively charged containing compound due to phosphate group. It gets attached to the positively charged amino acid of the beta chain of hemoglobin. As it occupies the central pocket of hemoglobin, so it is responsible for the unloading of oxygen from hemoglobin to tissues, and it is responsible for the maintaining buffering action due to phosphate group. So this is the function. So students, now coming toward the significance of this 2,3-bisphosphoglycerate. Now coming toward the significance of 2,3-bisphosphoglycerate. Students, this 2,3-bisphosphoglycerate is very important in anemia. In anemia, there is less level of the hemoglobin, and at that condition, this supplementary pathway carried out. This mechanism takes place. In case of anemia, this mechanism takes place, and 2,3-bisphosphoglycerate formation takes place, and it is responsible for the unloading of oxygen from hemoglobin to tissues. Okay, so in first case in anemia, it is very important. Next one is at high altitude. At high altitude, there is decrease in concentration of oxygen, and at that condition, this pathway takes place, and it is responsible for unloading of oxygen from hemoglobin to tissues. Student, this unloading of oxygen from hemoglobin to tissues, it is a very important function. This function is very important in case of anemia at high altitude. And third one is in case of fetal hemoglobin. This fetal hemoglobin having less affinity for 2,3-bisphosphoglycerate as compared to adult hemoglobin, but having high affinity toward oxygen. So therefore, there is placental transport of oxygen from the placenta to fetus. Okay. Now fourth one is in coronary obstructive pulmonary disease is very important. There's a less affinity of oxygen, so this pathway takes place, and there is production of 2,3-bisphosphoglycerate, and it is responsible for unloading of oxygen from hemoglobin to various other tissues. So students, in four cases, in case of anemia at high altitude, in case of fetal hemoglobin, in coronary obstructive pulmonary disease, it is very important. In anemia, there is less level of hemoglobin, so इस केस में क्या होगा कि फिर यही रेपोर्ट ल्यूबेरिंग साइकिल ही इसकी ये पाथवे क्रिएटर होगा आरबीसीज में, और क्या होगा हमें हमने भी पढ़ा है कि इट इज रिस्पांसिबल फॉर अनलोडिंग ऑफ ऑक्सीजन फ्रॉम हीमोग्लोबिन टू टिश्यूज एट हाई एल्टीट्यूड। जैसे ही हम बुलंदी पर जाते हैं ऑक्सीजन लेवल कम होता जाता है, तो उस सिचुएशन में फिर यही पाथवे ही चलेगा रेपोर्ट ल्यूबेरिंग साइकिल वाला, और 2,3 बिस्फोस्फोग्लिसरेट की प्रोडक्शन होगी एंड इट इज रिस्पांसिबल फॉर अनलोडिंग ऑफ ऑक्सीजन फ्रॉम हीमोग्लोबिन टू टिश्यूज। In case of fetal hemoglobin, there is a less affinity for 2,3-bisphosphoglycerate by high affinity for oxygen. So there is a placental transport of oxygen from placenta to fetus. And in case of coronary obstructive pulmonary disease, there is also less affinity of oxygen toward the various other tissues. So at that condition, this pathway takes place and it is responsible for unloading of oxygen from hemoglobin to various other tissues. So student, it was all about the Rapaport Leubering cycle, summarizing all. In this cycle, there is synthesis of 2,3-bisphosphoglycerate in RBC. It is very important. 2,3-bisphosphoglycerate is responsible for unloading of oxygen from hemoglobin to various other tissues, and it is responsible for maintaining buffer due to containing phosphate group. This unloading of oxygen from hemoglobin to other tissues, it is very important in 4 cases. First one is anemia at high altitude in case of fetal hemoglobin, and in case of coronary obstructive pulmonary disease. Students, if you have any query, you may ask in comment section, and keep watching and thank you. अगर आप इस वीडियो को लाइक कर वीडियो को आप पसंद आई है, लाइक भी करें, शेयर करें और सब्सक्राइब करें। थैंक यू।